Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Razan, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We have muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, because this is, after all, Ask the Expert, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. Thank you again for joining us today. It is now my pleasure to introduce our speaker, Gareth Meek-Brown from Lonza.
Thank you. Good morning, good afternoon, and good evening to everyone. I'm Gareth Meek-Brown, a lead scientist in process analytics, which provides analytical support both upstream and downstream process development activities at Lonza Slough, which is a process development and manufacturing site for drug substance produced by mammalian cell lines. Thank you for joining us for today's webinar, Product Quality Assessment using N-linked glycan analysis by LC-MS supports identification of commercially viable cell lines for biopharmaceuticals. Please note, we have a disclaimer for any forward-looking statements. I'll give a quick overview of the method, followed by a case study before we take questions. In Process Analytics Lonza, for mammalian development work, we use high-throughput methods where possible to streamline the analytical activities for process development. This webinar will focus on the Glycan application and how we've used Glycan data to select upstream process conditions and cell lines. Samples are generated from upstream process development coming from either standard or miniature bioreactors. The samples are then purified, either on board one of the robotic systems on the left, or as part of the N-linked Glycan preparation method on board our customized robotic system. We also have a multi-sample actor available for more complex purifications of small sample volumes in the milliliter range when required. After glycans are released, they are then available for analysis, either by UPLC or LC-MS, with an option for either rapid analysis suitable for general monoclonal antibody glycan profiles, or a 45-minute gradient suitable for tri- and tetraantennary structures. This diagram shows an example of a monoclonal antibody purification. However, for products with no affinity to protein A, an appropriate resin for a recombinant protein can be used for the initial purification. The onboard purification suitable for affinity resins, such as protein A, starts with resin equilibration followed by product loading. As it is an affinity resin, the non-bound, i.e. non-product material, can be washed away, allowing the product to be released in a separate step which generates a product with high purity, normally over 95%. An automated product protein concentration normalization then occurs on board before the samples enter the Glycan portion of the process. This is facilitated by the onboard plates reader, which is used as a spectrophotometer in a 96-well format, which allows the product concentration normalization to occur in an automated fashion. Once we have purified material, the product is bound to a cartridge and PNGase F is added, which cleaves the N-linked glycan at the asparagine site. The glycans are then dried out prior to labeling with a 2AB fluorophore for fluorescence detection by either UPLC or LC-MS. After the 2AB labeling step, the released glycans undergo a cleanup step which removes free 2AB label. The samples are now available for analysis either by UPLC or LC-MS. For LC-MS and UPLC, we use a Hillock column with fluorescence detection and Waters software. We have found that this screening method is suitable for both recombinant proteins and monoclonal antibodies. Here we have an overview of the workflow. We start with our fluorescence and MS data, and using a dextran ladder standard, we're able to convert retention time to glucose units using a retention time calibration, what we are then able to do is take the unknown peaks in the sample, which are then assigned a GU value, and we can use this to search against the database for hits. At the same time, the MS data is processed, and we have mass information for each unknown peak. This is also searched in the database to give a final proposed structure where the result is quantitated for relative percentage based on the fluorescence data, and the assignment is based on both the GU, which is a measure of relative retention time, and mass confirmation. Now let's move on to our case study. Our customer required us to produce a specified glycosylation profile for their biopharmaceutical, which in this case happened to be a biosimilar. We performed this work in 2 stages. For stage 1, we performed a scouting run to determine reactor conditions. And for stage 2, we analyzed cell lines using the selected reactor conditions to identify the top 12 cell lines. For stage 1, we performed a scouting run using modifications to our version 8 process in order to achieve the target profile as shown in red. The graph reports the mean results for each condition, conditions 1 to 6, which are modifications to the version 8 media. And then you can also see we have the control result as well. The control was produced by running the version 8 media without any modifications. Additional controls were also run, but this data is not shown. Modification 4, which is shown in grey, which you can see on the graph, was selected as the best modification based on a range of analytical data as shown in the blue boxes. So here we have cell viability, percentage purity, charge variance analysis, aggregate analysis, titer, and finally the topic of this webinar, Glycan profile. Now that we had the process from stage 1, we moved on to stage 2, selection of the top 12 cell lines. For this stage, we started with 24 transvectant pools. These were sorted into 531 clonal cell lines using automated colony identification, sampling, and picking. These were then ranked by octet for titer, and the top 90 clonal cell lines were selected for analytical screening, including full glycan analysis at 2 time points. That was 180 samples for glycan analysis. After analytical screening, these top 90 clonal cell lines were then run in larger bioreactors to better mimic the final process, and then were evaluated to further differentiate the top 12 clonal cell lines. Here we have a selection of the results from stage 2. You can see here we have the monospecies. Here we have the total galactosylation, total fucosylation, and then we also have some of the glycan structures, G0, G2, G0F, G1F, and G2F shown. Additional data was available, but for the purposes of presenting, I've trimmed the data down a little bit. You can see in red the clones which were selected for further evaluation. And finally, we have the results. Here's our results summary. We generated an N-linked glycan profile that met the customer target profile. We also met the additional criteria during the scope of this work, which included 3,420 individual analyses. The project time from transfection was 6 months. I'd like to acknowledge the following people for their work on this topic. Thank you. I'll now take questions.
Thank you, Gareth. So, for the first question, how long would the analysis for the case study have taken without automation? And how would the project time on slide 10 have been different?
So without automation, automation is the real driver here behind why we can actually screen so many clones. Without automation, screening large numbers of clones for product quality analysis just wouldn't be as possible. So to give an example, our traditional manual 2AB labeling process takes 3 days for up to 12 samples, whereas we're looking at about a day and a half for the automated purification for up to 96 samples and 2AB labeling for samples blank to controls. So you've got a massive time saving there using the automated method, which has also got efficiencies for the 2AB labeling as opposed to the traditional version of 2AB labeling. We're also using a much faster gradient for each sample. So once the sample is prepared, instead of running on the traditional method what would have been a 2-hour gradient, we've now got a 45-minute gradient that can be used for glycans up to triantenary and tetraantenary screening, or we have a rapid gradient of 7.2 minutes if we're just looking at biantenary screening. So biantenary screening would be limited to more simple profiles that you would associate with monoclonal antibodies. So overall, this screening, this many samples, just wouldn't have been practical for the customer, and there would have been a huge cost associated if the customer did decide to screen this many samples using a traditional, more manual, lower throughput method.
And does the released glycan need to be separated from the protein prior to 2AB labeling?
Yes. So what we have here is N-linked glycan analysis. Another type of glycan analysis is O-linked glycan analysis. With N-linked glycan analysis, it's the enzyme PNGase F has been available for a while, which allows glycans to be cleaved quite cleanly so they can be 2AB labeled with a fluorophore, because glycans themselves won't be detectable using a fluorescence method. So you really want that fluorophore to really boost that signal. But if you have O-linked glycans, there aren't actually any commercially available enzymes that can cleanly cleave off the O-linked glycan. There are some alternatives, but I won't go into these so that we stay on topic.
And how do the results compare to traditional QC batch release methods?
So we have used this method to support a manufacturing investigation. So in order to provide justification of why we were using this method, what we did is we analyzed some samples that had been analyzed using the batch release method, and we used those same samples here for the screening method and compared them. So in this particular example, the only 3 glycans that were concerned with was G0F, G1F, and G2F. And for those glycans, we saw only up to a 0.2% difference between the data. So this is data for our Lonza batch release method, which in this case was performed at another site, as we've got many sites globally, and the screening method performed here in the UK.
And are there any limitations of using the NIBRT library, and how confident are you on the proposed structures?
So the NIBRT library, the 2AB library, is a very good library, and there are over 300 structures that we can screen against. However, what we've found for mammalian cells is that you have a lot of, when you start to get to the more complex profiles with triantenary and tetraantenary, those structures aren't in the current NIBRT library that's available. And so for that, we've actually had to do our own work to provide information on the triantenary and tetraantenary structures. However, it depends on what purpose you're using the method for. If you're using screening for high throughput, what we've done is absolutely fine. But if you want to go into something like reference standard characterization or batch release testing, then you might want to do further confirmation using things like exoglycosidases to confirm unit, subunit confirmation, and linkage confirmation.
Okay. Well, we thank you very much for your talk today, Gareth, and we thank you for joining us.
Thank you.
As a reminder, this webcast will be made available in the multimedia section of the Bioprocess International website for on-demand viewing. Thank you.
Thank you, everyone.